Okay, so for this topic today, it's actually very it's very short. Sekejap je kita dah boleh habis this lecture. So actually it's also a recap from apa you belajar masa form 5 dulu. So tak ada pertambahan yang sangat obvious sangat, but you have to know nak bezakan antara meiosis dengan mitosis, bezakan antara meiosis 1 dan meiosis 2, function of meiosis, kenapa kita kena ada meiosis, and then the chromosome structure. Okay, we'll go into detail on all of that. Okay, nanti. So it will be from chapter 10 of your Medi textbook. Okay. So the outline, so this is the only subtopics we'll be covering for this syllables. We have 4 from this chapter. Okay, 10.1, which is the overview of meiosis, and then we have 10.2, which is the genetic variation. 10.3, we have the phases of meiosis. And lastly is the 10.6, which is the changes in chromosome number and structure. These are the subtopics that you have to concentrate on for our syllables. Okay. So topic 10.5 yang the cycle of life, and also 10.4. 10.4 tu actually masuk sikit, cuma kita tak go into detail masukkan dia sebagai satu subtopik. You have to also know 10.4 tu meiosis compared to meiosis macam mana. But I included that into my slide as well. Okay. So learning outcome. You have to know by the end of this lecture, you kena tahu how to contrast haploid and diploid chromosome number. Explain how crossing over contributes to genetic variation. Apa beza meiosis ni dengan mitosis? Mitosis tak ada crossing over. Meiosis ada crossing over, and why? You have to know that. And then contrast the changes in chromosome number, genetic variability, and number of daughter cells between meiosis and mitosis. Okay, so meiosis, why we are and who we are. Why do we need meiosis? Anybody can tell me? Why do we need meiosis? Mitosis untuk apa? Meiosis
Formation of gamete.
Okay, production of gamete, good. Okay, satu lagi. Kenapa? Why is it important?
Nak ada variation.
Variation, that's very good. Yeah, betul. Sebab dalam meiosis, 2 important steps that you have to remember. Satu is the crossing over, synapse. Lagi satu is the independent assortment, yang tak ada dalam mitosis. Okay, that creates genetic diversity. That's why we need meiosis. Okay, lagi satu is also to reduce daripada diploid ke haploid. Okay, half the numbers. What happens if we don't reduce the chromosome number? Imagine, okay, from your kata ni dah berapa generation dah. One generation produce berapa-berapa, and then next one dia akan double lagi, next one akan double, dia tak half. Bila double, you fertilize, so jadi lagi banyak. So your cells will be full of chromosome. So tak boleh kalau macam tu, it can't function. Apa ni? But every species have their own chromosome number. So bila is half gametes kan, fertilize, dia akan jadi that chromosome number again. Okay. So overview of meiosis. Okay. It is only used for sexual reproduction. Asexual reproduction tak ada meiosis. Okay. And then chromosomes are replicated in the S phase of interphase. Dia ada sama juga macam mitosis. It has the interphase. Okay. And then it's half prior to fertilization. So the parents are diploid. And meiosis produces haploid gametes. So haploid cell contain a single cell of chromosome. So if, this is what happened. If there were no reduction of chromosome, kalau tak ada meiosis, the number of chromosomes would double each generation. Now your cells will be just full of chromosome. Okay. So gametes fuse in fertilization to form a diploid zygote. Okay, haploid and haploid, dua jadi diploid. N jadi 2N. So the zygote becomes the next diploid generation. So if events in meiosis go wrong, gametes contain the wrong number of chromosome. Okay, yang ni yang akan jadi banyak lagi penyakit. Contohnya macam Down syndrome, Turner syndrome, Klinefelter. Okay. So, okay, this, if you see the GIF here, is meiosis. Okay, tak apa, we'll go into detail nanti. Okay, so chromosomes of the same type. So, if you refer on page 167, you have to know the term. Bila nak guna term homologous pair, bila nak guna term, homologous chromosome, bila nak guna term sister chromatic. Okay, selalunya bila kita cakap pasal meiosis 1, we refer to homologous chromosome. When we talk about meiosis 2, we talk about the sister chromatids. Okay, so please understand, homologous pair, ni chromatid, and this is sister chromatid, merah dengan merah. Okay, this is non-sister chromatid, merah ni dengan this sister chromatid, non-sister chromatid. Okay, yang merah dengan merah is called sister chromatid. Kena sama. Ni lain, jadi non-sister chromatid. Okay, you have to know where are the centromere and where are the kinetochores. Okay. So, homologous chromosomes have genes controlling the same trait at the same position. So, gene occurs in duplicate, satu daripada mama, satu daripada your papa. Okay, so many genes exist in several variant forms in large population. So homologous copies of a gene may encode identical ataupun different genetic information. Contohnya macam the copy of a DNA that brings about the trait of hair color. Okay, katakanlah you dengan adik-beradik you. Some maybe ikut mak rambut hitam. Some of your sibling maybe ikut your dad punya hair color. And also the type of hair. Wavy ke, straight ke, your parents curly and then straight, tiba-tiba you dapat wavy. So all that plays important roles. Meiosis plays important roles untuk control all that traits. Okay. So alleles. So the variants that exist for a gene are called alleles. So an individual may have identical alleles for a specific gene on both homolog. So bila identical, we call that as homozygous. Contohnya capital A and capital A, ataupun small letter A and a small letter A. So that is homozygous, dia sama, identical alleles. So bila it is not identical, it differs, we call that as a heterozygous. A besar, A kecil. Okay, nanti you have to understand what are alleles, sebab nanti bila next week we'll go into Mendelian inheritance, kita akan buat banyak crossing. Crossing, you have to remember homozygous macam mana, heterozygous macam mana. Homozygous, contoh, saya bagi contoh kalau A punya allele, A besar, and A kecil, homozygous. Kalau heterozygous, a mixture, different, okay, A besar and A kecil. Okay. Okay, so next we go on into meiosis itself. Okay, this one. Okay, meiosis 1, chromosomes are replicated prior to meiosis 1. And then each chromosome consists of 2 identical sister chromatid. Dalam prophase 1, we have the synapsis. So the homologous chromosome pair up in synapsis. Okay, remember the term, meiosis 1, homologous chromosome. Okay, don't use sister chromatid here masa synapsis ni. Memang salah. A lot of students get confused between chromosomes and chromatid. Okay, don't get confused, please. So chromosomes may recombine or exchange genetic material. So that is synapsis, crossing over, another word for it. Okay. So homologous pairs align themselves against each other, side by side at the metaphase plate. Homologous pair. Okay. Two members of a homologous pair separate. This is anaphase. And then each daughter cell receives one duplicated chromosome from each pair. And the chromosome number is reduced from diploid to haploid. Okay, so in meiosis 2, DNA is not replicated. Okay, antara meiosis 1 and meiosis 2, they have a phase called the interkinesis. It's a resting phase. Okay, so DNA, but in interkinesis, dia tak sama macam interphase. Interkinesis tak ada replication. Okay, it's just a resting phase and also the formation of the microtubule central balik daripada satu nak jadi dua. Okay, nanti I'll show you nanti lepas ni, okay. So DNA is not replicated between meiosis 1 and meiosis 2. That is interkinesis dia. Nah, sister chromatids. Okay, bila dah guna meiosis 2, bila meiosis 2 you guna the term sister chromatid. Okay, sister chromatids separate and move to the opposite poles. And 4 daughter cells contain 1 daughter chromosome for each pair. So each daughter chromosome consists of a single chromatid. And daughter cells ni adalah haploid. So we'll go into detail on each of the step. So this is the overview of meiosis. Overview, daripada awal we have the meiosis 1. Separate jadi 2. And then we have meiosis 2. Last kali kita ada 4 haploid daughter chromosomes. Okay. So genetic variation is essential for a species to evolve and adapt in changing environment. So I said bila meiosis ni, the two important event that happen in meiosis is the crossing over. Okay, this is in prophase between homologous chromosomes and independent assortment. Yang ni berlaku dalam metaphase. Okay, prophase and metaphase. So they happen in meiosis 1. So prophase 1 and metaphase 1. Okay, crossing over is between homologous chromosome and independent assortment also between the homologous chromosomes. So what is crossing over? Synapsis. So it involves exchange of genetic material between non-sister chromatid during meiosis 1. Okay, crossing over. So at synapsis, nucleoprotein lattice called the synaptonemal complex appears between the homolog. So this holds the homolog together, aligns the DNA of non-sister chromatid, and then allow the crossing over to happen. This is the chiasmata of non-sister chromatid, the point of the crossing over. Okay. So homologs and separate and distributed to different daughter cells. So daripada you nampak yang ni A, B, C, D merah and A, B, C, D biru ni kan. Dia dah bergabung because there is the crossing over has happened. So this is called synapsis. Crossing over has occurred. Okay. So another important event is the independent assortment. Okay, independent assortment, you have to know because nanti dalam Mendelian inheritance, dia ada law of independent assortment. Okay, bila law of independent assortment, you refer back in meiosis. It happens in the meiosis. So when homologous chromosome pairs align at the metaphase plate, okay, bila homologous chromosome, you tahu dia metaphase 1. Okay. Metaphase 1. So they separate in a random manner. They are independent, very random manner. Okay. This causes random mixing of blocks of alleles into gamete. Okay. Dia ada dua orientation. Sama ada it goes either towards the maternal or the paternal maternal or paternal homolog may be oriented towards either pole. So dia boleh dua orientation, satu to the north, satu to the south. Okay, that's why dalam the formula untuk nak calculate the diversity or the variation of a certain species, contohnya bila kita ambil human, okay, orientation dia dua, south and the north, dua. So atas ni, kalau yang ni 3, kita dalam human is 23. So you calculate 2 to the power of 23. So how much you get? So contohnya kalau yang ni the simple explanation dekat sini, possible chromosome orientation for a cell with 3 pairs of homologous chromosome is 2 to the power of 3 and kita dapat 8. So these are the 8 combination that you can get kalau ada 3 pairs of homologous chromosome. You get 8 different combination. Okay, this is called independent assortment. Okay, so fertilization pula. Union of male and female gametes. So as I said, in humans, dua tadi tu, orientation, south and north, dua to the power of 23 gametes kan? And then kuasa dua because we have the male and the female during during fertilization, we have both gamete, male and female. So this is the number. Saya nak sebut pun saya dah tak tahu dah berapa ni. This is the possible variation that can happen in the human. So contohnya saya satu, you punya satu. So in order for you to be sama dengan another individual yang lain, it is very 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 rare or very impossible at all. Sometimes ada scientists believe sebab dia buat genetic linking and mapping, dia kata ada, it does happen, cuma very very rare. Okay, so imagine if crossing over occurs only once pun dah berapa. Okay, genetic difference I got. So crossing over may occur several times in each chromosome. So that's why you are special. You are not the same as your friend. Generally. Okay. That's why we masing-masing semua ada our own DNA that is very different from our friends. So that's why you are unique. You are special. So significance. Kenapa kita kena ada genetic variation? So the significance of genetic variation. Satu, kalau asexual, beza dengan sexual. Asexual, dia produce the same, identical cloning. Okay, clone. Kalau sexual pula, kita ada genetic recombination. So bila ada genetic recombination, kita ada variation. So bila ada variation, it produce a lot of advantage over the asexual reproduction. Contoh, some offspring may have a better chance of survival. Contohnya, dia ada fur atau reduced body fat. Okay, kalau clone, you clone satu benda tu, less fur lah dia sampai generation ke berapa bawah. Kalau genetic, kalau sexual reproduction, it can be altered. Maksudnya it goes according to it adapts according to the climate atau to the environment. Contohnya less fur, tiba-tiba boleh jadi fur lagi banyak, evolution kan? Less fur lagi banyak boleh jadi fur yang thick. Okay, so those are all genetic recombination that can happen during sexual reproduction. Asexual reproduction, you have to know it is directly clone. It's sama. Okay, so the phases of meiosis. So we'll go on into phases of meiosis. So we'll be in subtopic 10.3. So what happened? I'm sure you guys know already what happens in meiosis 1 and meiosis 2, but I'll just do a quick recap. So prophase 1, what you have to know is the important event of synapsis or the crossing over. Okay? So before meiosis starts, memang dia ada interphase as well, macam mitosis. So interphase and then prophase 1. Prophase 1, the key event that happen is synapsis. So bila ada synapsis or the crossing over already, the synapse homolog are referred to as bivalent. Two homolog yang dia dah gabung, dia dah crossing over has occurred. We call that as a bivalent. We refer that as a bivalent lepas tu, ataupun tetrad for chromatic. Okay. So next, after prophase 1, we have the metaphase 1. Metaphase 1 ni is the independent assortment. Okay, dia line up at the metaphase plate. Yang ni dia akan berlakunya independent assortment. So by bivalence ni kan, yang tadi tu, are aligned at spindle independently of one another. So after metaphase 1, we have the anaphase 1. Anaphase 1 is when homologous chromosomes of each bivalent separate from one another. Okay, they go towards the pole. So katakanlah di North Pole, so this is the South Pole. So the homologous move towards opposite poles and sister chromatic do not separate. So yang ni adalah homologous chromosome. The sister chromatic do not separate masa meiosis 1 ni, okay? punya anaphase. So each still a duplicated chromosome with 2 chromatid. Okay, cuma there is a reduction of chromosome number from 2n ke n. Okay, so telophase 1 daughter cells have one duplicated chromosome n from each homologous pair. Right? So after telophase 1, okay, this is important. You have to know beza antara mitosis and meiosis. Mitosis tak ada interkinesis. Okay, interkinesis only happen between meiosis 1 and meiosis 2. So two haploid daughter cells, each with one duplicated chromosome of each type, is in interkinesis, and interkinesis is similar to the mitotic interphase. Cuma it is shorter. Dia sekejap je. It's like a resting phase. And DNA replication does not occur. So what happened during interkinesis ni? Actually kalau you perasan, central kat sini ada berapa? Dia ada satu kan? Centrosome ni. Okay, satu. So when you see prophase 2, dia ada dua. Okay, nak jadi spindle fibers again. Jadi macam ni. Okay, so that is why we need interkinesis. For the formation of the microtubules, untuk nak form the spindle fibers to continue meiosis 2. Okay. So, starts of prophase, so dia dah ada dua centrosome or dua centrioles. Okay. The chromosomes still condense. And then, it will align metaphase 1. So, dia is no longer in homologous pair dah. Okay, dia tak ada kawan dia lagi satu dah. Right, so anaphase 2, centromere dissolves and sister chromatids separate, become daughter chromosome. So telophase 2 and cytokinesis, lastly they akan produce 4 haploid daughter cells that are genetically unique. Okay, so gametes containing a mixture of maternal and paternal genes already. Okay, kalau you tengok tu, because of the crossing over occurs and also the independent assortment, so you will get different variants. You get 4 haploid daughter cells that is different from one another. Okay. Okay, so let's go through this again. Meiosis 1. Okay. Kalau you tak nak baca pening-pening banyak-banyak sangat the details, you can just refer to this figure. This figure is on page 173, which is figure 10.6. So you will know what are the stages or the phases in meiosis 1, what happened, and meiosis 2. Okay, daripada 2 ni, 2 go on into meiosis 2 pula. Jadi 4. Okay, meiosis 1, you have to label, kalau dalam exam, you tulis meiosis prophase saja. Crossing over happen at prophase. Salah. It has to be prophase 1. Sebab meiosis ada 2. You have to be specific. Okay? Alright. So, the differences. Macam ni. Mitosis. Attack of the clone. Okay, yang ni meiosis. We dare to be different. Nampak tak? Dia punya variant semua lain. Ni purple sama juga. Purple je. Ni daripada hijau, dia boleh jadi biru, dia boleh jadi merah. Contohnya lah. Okay? So this is beza antara mitosis and meiosis. Okay, so in meiosis, alah terbalik pula yang ni. So this should be here, mitosis. Sebab gambar ni mitosis kan, so abaikan je lah. So just have to refer to this meiosis and mitosis. So this meiosis requires 2 nuclear division, meiosis 1 and meiosis 2. Mitosis pula only 1. Dia tak ada mitosis 1, dia tak ada mitosis 2. Okay. And then chromosome synapse and crossing over occurs in meiosis, while it doesn't in mitosis. And then it halves the chromosome number. Mitosis preserves the chromosome number. Meiosis produces 4 daughter nuclei, and mitosis produces 2 daughter nuclei. And then meiosis produces daughter cells that are genetically different from parent and also from one another. Kalau mitosis, sama. Produces daughter cells that are genetically identical to parent and also to each other. Okay, so meiosis is only used in sexual reproduction. Gametes, okay, always refer to meiosis ni as gametes. They only use for sexual reproduction. Kalau mitosis, they are used for asexual reproduction and also for growth. Anything lah, growth ke, cut ke, okay, all that are mitosis. Okay. So, okay, this is the overall apa ni, table juga. You can refer that on page 175. Okay. So, this is comparing between meiosis 1 to mitosis and also meiosis 2 to mitosis. What happened? Meiosis 1, macam tadi jugalah, is lebih kurang. But we repeat it again. Prophase 1, ada pairing of homologous chromosome or the crossing over. Mitosis tak ada. Okay. Metaphase 2, metaphase 1, the bivalent kan, is at the metaphase plate. Bivalence ni, you only refer bivalence because there is crossing over. Baru you refer it as a bivalent. Okay. Kalau tak, we refer it as a duplicated chromosomes atau homologous chromosomes je at the metaphase plate untuk mitosis. Anaphase 1, homologs of each bivalent separate and duplicated chromosomes move to pole. Anaphase mitosis, they separate and becoming daughter chromosomes that move to the pole. Okay. Telophase 1, dia 2 haploid kalau meiosis. Kalau mitosis, dia jadi 2 diploid. Daughter cells identical to the parent. Kalau meiosis is not identical to the parent cell. So this is meiosis 1. In meiosis 2, tak ada pairing. There is no crossing over occurs in prophase 2. Okay. And dia actually prophase meiosis 2 ni is quite similar to mitosis. Kalau you nak refer dia lah, sebab there's no crossing over, dia tak ada independent assortment happening, betul-betul homologous chromosome dekat metaphase 1. Okay. So metaphase 2, haploid number of duplicated chromosomes at metaphase plate. Metaphase mitosis, the diploid number of duplicated chromosomes at metaphase plate. Anaphase pula, sister chromatic separate becoming daughter chromosomes that move to the pole. Sama juga dekat mitosis, sister chromatic juga separate. Okay, cuma beza dia meiosis 2 dengan mitosis ni haploid number and also the diploid number. Telophase 2, 4 haploid daughter cells not genetically identical produced. Kalau telophase dekat mitosis, 2 diploid daughter cells identical to the parent cell. Okay. So this is the same thing again. Cuma is in figure. I think this is for you, you lagi senang nak faham apa jadi dekat meiosis 1, apa jadi dekat meiosis dan apa jadi beza dia dengan mitosis. So the main difference is the alignment dekat sini. Ni bivalent. Okay, bivalent homologous pair. Yang ni dia tak bivalent. Dia only duplicated chromosome. Dia tak ada crossing over. You tengok dia merah biru je. Okay. And then dia apa ni, anaphase. So, dia tarik the whole thing. Dia connect to the kinetochore sini, dia tarik the whole thing kalau meiosis. Kalau yang dalam sini, dia connect to the kinetochore dekat the sister chromatic. So, sister chromatic yang separate. Itu beza antara meiosis and mitosis. Right? So, changes in chromosome number and structure. So, we go on into the last subtopic, which is 10.6. Okay, 10.6 ni, meiosis almost always proceed normally. Okay, when it's normal, kita cakap, we call that as euploidy. Euploidy is the correct number of chromosome in a species. Kalau aneuploidy, dia ada change ataupun kita dah panggil dia sebagai apa ni, dia dah pernah go through any non-disjunction. Okay, which is failure of chromosome untuk nak separate. So aneuploidy, we call that aneuploidy and euploidy. Euploidy is the correct number. Aneuploidy is not. Okay. Aneuploidy ni ada dua. Okay. The non-disjunction can occur in meiosis 1 or meiosis 2. Sama ada primary non-disjunction ataupun secondary non-disjunction. So, this non-disjunction may result in whether gain or loss of chromosome. Sama ada dia tolak satu ke ataupun dia akan tambah satu. Kalau tolak satu, we refer that as monosomy. Okay, monosomy. Only one of a particular type of chromosome. Kalau trisomy, we have 3 of a particular type of chromosome. Okay, there. This is a primary non-disjunction. Dia occur during meiosis 1 punya step tu. Okay. Primary, atas. So this is secondary non-disjunction. So what happen during non-disjunction? Sepatutnya yang ni homologous pair ni satu dekat sini, satu dekat sini. Tapi dah jadi dua-dua masuk the same cell. So this is primary non-disjunction. Dia akan hasilkan, bila dia separate, jadi 2n plus 1 and 2n plus 1 and 2n minus 1 and 2n minus 1. So this is monosomy, this is trisomy. Okay. Dia salah satu je dekat primary dekat atas ni, tapi salah sampai habis, dia replicate sampai habis. Salah. So kalau secondary non-disjunction pula, dia mula-mula okay, dia separate normally. Tapi bila dekat meiosis tu, dia dah salah. Okay, what happened here? Dia separate, okay, this one okay, this is normal. Yang ni pula termasuk satu dekat sel ni. So what happened? These cells are normal. Dia jadi normal diploid cell. Okay, and yang ni pula yang salah. So 3 and 1. So trisomy and monosomy. So I would like to ask you guys punya opinion. Yang mana satu you rasa lagi bahaya? Primary non-disjunction or secondary non-disjunction? Rasa-rasalah. Yang mana?
Tangan kamu.
Second.
Primary.
Primary.
Primary, betul. It's primary. Sebab secondary, you can still get normal. You can still get a diploid. Dia tak jadi semua. Kalau primary, kacau dekat sini, semua bawah ada kacau. Trisomy and monosomy. Kalau yang ni, dia still boleh develop some normal diploid. So, ada satu sel je yang trisomy and also monosomy. Kalau primary non-disjunction, dia terus semua tak akan jadi normal diploid cell. Okay? So, that is the main difference between primary and secondary non-disjunction. Okay. So, trisomy 21. Ni kita selalu dengar. Apa ni? Selalu kita call that as a Down syndrome. Okay, ada extra chromosome dekat chromosome 21. Ya, 3. 2n plus 1. Okay, trisomy. Kan tadi kan, kita refer to 2n plus 1. So, ni, sepatutnya dia 2. Tapi dia ada extra. So, yang ni, kalau you refer, I'm sure you know how Down syndrome individuals are like. They orang have wide gap between first and second toe. Dia ada macam flat face. They have stubby fingers. Tu kalau you perasan kan, all these special kids, Down syndrome individuals, mostly dia punya physical punya ni sama. Dia tengok, dia ni macam sama dengan kawan-kawan dia kan. You can't really tell them apart sangat because of the characteristic ni. An extra chromosome at chromosome 21, so dia akan develop this characteristic. Dia punya eyelid fold, dia ada flat face, dia ada stubby fingers. So kalau you refer to page 166 yang Brandon ni, okay, that's how a Down syndrome look like. Okay, Down syndrome is a trisomy. Trisomy maksudnya dia ada extra 1. Okay, selalunya kenapa jadi? Well, we can't really say it's definite because selalunya orang blame the mother kahwin lambat ke, you know, because the longer the usit ni stored dalam badan, the greater the chance of a non-disjunction occurring. Tapi wallahu alam, we can't really say that is the main cause of Down syndrome. Sebab ada je yang parents yang kahwin biasa umur 20 plus and still ada anak Down syndrome. Okay. So another condition, Turner syndrome. Okay. Turner ni female with a single X chromosome. Dia jadi X kosong, dia hilang satu. 2n minus 1. Okay. Female with single X chromosome. Short, broad chest. Dia jadi macam lelaki dah. Under widely spaced nipple. They can be of normal intelligence, tapi dia kena be on hormone therapy. Okay. So these are normal female. Ni kalau Turner syndrome. Okay, dia short. Dia macam stunted sikit. Okay, that is X0. X0. Kalau Klinefelter syndrome, XXY. Okay, XY is male. Tiba-tiba ada extra X. So, dia develop female punya karakteristik. So, kalau you tengok gambar dekat sini, this is a male. Tapi dia ada larger breast and also weaker bone, dia ada lower energy level, dia ada smaller penis and testicles, dia delay in puberty. These are the sign of Klinefelter syndrome. Maksud dia ada extra female punya karakteristik, extra X kat situ. Okay. Okay, tadi Turner missing chromosome X dekat sini. So dia jadi X kosong. X0. Tadi kan yang ni, Turner syndrome, X0. So this is Turner syndrome. Ni Klinefelter, dia ada extra. Sepatutnya XY saja kalau male, tapi dia ada extra chromosome X dekat sini. Okay, so that's why dia display ada extra female characteristic. So these are example of Turner syndrome and Klinefelter syndrome. Okay, last part here.
Doktor,
Yeah,
sorry to interrupt.
yes,
But,
okay.
Maksudnya yang Klinefelter syndrome ni yang macam Hunsa tu ke?
Tak, Kunsa tu lain, dia develop an organ, betul tak? Yang ni dia tak develop organ, cuma dia develop, dia macam jadi dia punya body tu dia develop breast, betul tak? Dia tak ada an extra organ. Kalau Kunsa dia ada 2 organ, betul tak? Klinefelter is not Kunsa.
Oh, I see.
Okay, kalau Kunsa dia ada dua-dua. Yang ni dia tak ada dua-dua, dia cuma underdevelop je dia punya testis dia and prostate, and dia ada breast overdevelopment. Okay, dia kalau you tengok dia macam, kalau yang pondan, sorry to say, yang pondan tu dia cucuk untuk nak jadikan breast besar semua kan. Ni dia naturally. Cuma organ dia tak silap saya, Klinefelter tak ada. Dia tak ada 2 organ. Dia still sama. Organ yang sama. Sebab kalau Kunsa, dia ada 2 organ and some ada rahim. Ada apa ni? Ada rahim lah. Tapi yang ni tak ada. It's just the general characteristic dia je. I'll correct if I'm salah nanti. But that's what I understand lah. Klinefelter is different than Kunsa. Okay? Anybody know? Betul ke ni? Tak silap saya tak ada. Kejap. Let me Google.
Doktor,
Ya.
jadi kalau Hunsa tu sebab bukan changes in chromosome number ke macam mana?
Actually kalau Kunsa tu more to change, bukan Kunsa. Yang tu saya tak berapa nak tahu, yang tu I have to find out. Okay. Nanti I let you guys know sebab saya tak adalah detail sangat study on Kunsa punya ni, but I'll read about that nanti later and I'll let you guys know dalam group, okay? Right? So nanti saya tanya juga, is Klinefelter selalunya tak, dia tak ada dua organ. Dia only satu je. Okay? Okay, so lastly is the changes in chromosome structure. Okay, so that is on page, sama juga yang tadi tu. Cuma changes in the chromosome structure is on page 180. So yang ni pun you dah pernah belajar before. Ada deletion, ada duplication, ada translocation, ada inversion. So kalau deletion, contohnya dalam sini A, B, C, A dia dah terkeluar. Okay, so that is deletion. Kalau duplication, A, B, C, D, E, F, G, dia duplicate D, E. So this is duplication. Kalau inversion pula, inversion dia break dulu. Dia break dua ni, C dengan D, lepas tu dia reinsert balik. D dengan C kat sini, terbalik. Inversion. Okay, lagi satu, last one is translocation, which is dia move to another non-homolog punya chromosome. Macam ini, QR dekat sini, dia dah move ke sini, GH dah move ke sini. So that is translocation. Okay. So human syndrome yang berkait dengan deletion and also translocation. Kita ada Williams syndrome kalau deletion. Loss of the end of the chromosome 7 which contain gene for elastin. Okay, children have turned up nose, but nose dia. Okay. Dia macam wide mouth, a small chin and large ear. Tengok dia punya ear dia. Okay, so this is contoh deletion syndrome. Another one is translocation syndrome. Selalunya kalau translocation ni kita banyak libatkan blood. Okay, macam leukemia and all. So selalunya akan lead to cancer lah kalau bila ada translocation syndrome ni, sebab dia dah alter the whole of that. Kalau you tengok ni, dia dah jadi baru. Coding dia pun baru dah. Okay. Translocation. Selalunya bahayalah translocation. Sangat-sangat bahaya. Okay. Alright. So that is all for meiosis punya topic for today.